Thank you very much to Fatigatio and also to Birgit to insist to give a presentation today. I actually decided in the beginning that I probably would not give a talk this year, but Birgit insisted. The reason for me not to have interest this year was the recent COVID-19 pandemic. We had really a tough time. For many weeks, we did not go to the lab, and then we were only allowed to go to the lab for 3-4 weeks. Students were not allowed to work for many months also. So it was extremely tough. Right now we are back in lab, but still we have restrictions in working with blood samples from patients and many different other type of restrictions. So the last 5-6 months had been very tough on us from the research point of view. But still, today I'm going to share some of my old data as well as some new data which is not yet being published. To give you an idea what we are actually doing and what we are planning to do in the future. So my talk is today mitochondrial dysfunction in ME/CFS. I would like to start my talk by thanking the partners who helped us providing samples and also time-to-time discussions. And so Carmen from Berlin, you have heard her today. Uta Behrens from Munich, Thomas Harrer from Erlangen. These are the 3 clinical partners from within Germany. And I am greatly thankful to all 3 of them. And I'm also thankful to Robert Navio from University of California, San Diego. He's a great man. It's an honor to work with him and share scientific excitement in everyday life. And I'm looking forward to have more publications, great work with him. I would like to thank the very few number of funding agencies who came forward and helped us in a tiny way, but very timely help, which allowed us to work on this mystery disease. And recently we got money from Austrian funding agency also to look into ME/CFS, which is extremely helpful. And last but not the least, I would like to thank the patients, ME/CFS patient community. And recently they did the GoFund ME/CFS fundraising, which not only tells us that they have faith and trust in our work, also gives us a little more hope financially to go ahead. So thank you all of you for your help. So I would like to start my talk with 2 very well accepted facts that ME/CFS is a multifactorial disease. So some of the researchers, clinicians believe that this is an autoimmune disorder. Some will say that it's an infectious disease. Some will say that it is because of the infection with herpes viruses, EBV, HHV6/7. Some will say that it is because of the infection with enteroviruses and retroviruses and things like that. They all are right, probably. I would not decline anyone's research here, because I myself also feel that it's a multifactorial disease. It's not, every patient is different. Every patient has a different profile which we cannot match with another patient. This is something very hard that I have learned in the past few years. And that's why it's very hard to find a unified biomarker for this disease. But I personally feel that it might be possible that we can tackle the clinical symptoms of this disease by understanding the cellular factors that affect the mitochondrial health. And the mitochondria is very key to this disease. It is also key to many other diseases, but this disease is very special here. So I would like to talk a little bit about our work on mitochondria. So mitochondria is a very dynamic structure in the cell, and it changes its structure. It's not like a stable or fixed architecture. It changes from a very fragmented form to fission, small mitochondrial form from time to time, but this change is very necessary. But it's very interesting that different pathogens, viruses, bacteria, and different type of pathogens, they target mitochondria. Whenever they infect the cell, one of the most important structure within the cell that they would like to change or hijack is mitochondria, because mitochondria not only provides them the nutrients, the energy for them to grow, replicate, but it also, another beautiful function of mitochondria is innate immune response. It protects the cell from incoming viruses and other pathogens. And because of that, the pathogen has to survive inside the cell. And that, for that reason, they have to somehow change the mitochondria so that they have a better chance of survival. So the pathogens, they target mitochondria, but once the pathogen targets mitochondria, the cell realizes that something wrong is going on. And then it changes the mitochondria itself. The cell changes the mitochondria itself, or the subsequent signaling pathways to tell itself, the cell which is being infected, as well as the cells in the surrounding, that there is something wrong going on in the nearby. So get prepared for it. So we are fascinated in understanding how this change from pathogen changing mitochondria to mitochondria changing its response to the cell affects pathophysiology, and in particularly in ME/CFS, how it happens. So, as you know, that my lab focuses on one of the pathogen, which is HHV6, human herpes virus type 6. It's a herpes virus, one of the 8 different herpes viruses that infect human beings. And we have recently published that one of the key feature of HHV6 infection and the reactivation is that when HHV6 reactivates, it fragments mitochondria. So when I say fragments mitochondria, you can differentiate between that. This is the normal filamentous form of mitochondria, which is a healthy form of the mitochondria. And once the virus reactivates inside the cell, you can see that the mitochondria acquires a more punctated structure, a circular spherical structure, and stays in this structure for a longer time, which is the worst part of the cell. Converting to this structure is not the worst part of this. But if it stays in this structure, that's not good for the cell, okay? And we also recently published and showed that this phenotype of fragmentation of mitochondria is actually possible to be transferred. That means the cell in which virus is there the mitochondria definitely fragments. But if we take the culture supernatant from these cells and put it on healthy cells, they also fragment mitochondria. So the structure of the mitochondria changes. It's just a quantitative mitochondrial surface area measurement where you say that the cells which are treated with the drug called TSA, trichostatin A, to reactivate the virus, they only induce a smaller mitochondria. But the same cells without having the virus, even if they are treated with TSA, they don't do the same thing. So mitochondrial fragmentation can be transferred from one cell to the other. Together with Bob Navio, we did a big project recently with the help of funds generously provided by the patients and the other private donors. We did a metabolomic study to understand what are the metabolic changes happening in the culture supernatant where the virus is being reactivated and not reactivated. I'm not going to discuss the results today because this is a joint effort from Bob's lab and my lab, and we would like to come to a common conclusion, and then we'll do. But we have very fascinating results, and on the basis of results, we have also started to try some metabolic mediators which are decreased to add to the cells and see that if the mitochondrial fragmentation can be recovered. So it's a very fascinating story. But here I'm going to talk about something else. So as you know that I have been talking about it many times, that there is a small microRNA which is being produced by HHV6. It's called microRNA U14, and this causes the mitochondrial fragmentation. That's why HHV6 is unique. No other virus is known to produce such microRNA which can cause mitochondrial fragmentation. At least we don't know. So here we can show that if the cell is healthy cell is transfected with the small RNA, which is not microRNA U14, you see very nice mitochondria over here, filamentous mitochondria. But the moment we transfect the cells or put this microRNA U14 into the cells, you see these fragmented spherical mitochondria. Now, this is very unique for HHV6, but this is not unique for viruses. If you look into other herpes viruses, for example, varicella zoster virus, it produces immediate early protein 63, i.e. 63 protein, which also targets mitochondria and fragments it. If you look into human cytomegalovirus, it produces UL37 protein, which also fragments mitochondria. If you look into EBV, it also produces a membrane-associated protein, LMP, late membrane-associated protein, LMP1, which also fragments protein. These are some of the examples. So many herpes viruses actually fragments mitochondria. So my favorite is HHV6, but this doesn't mean that HHV6 is the only virus causing it. What I'm trying to tell here is that it might be possible that many viruses have many different strategies to fragment mitochondria. But now what happens when mitochondria is fragmented? If it is fragmenting continuously, then it leads to senescence, mitophagy, and if localized reactivations occurs, it can cause inflammation, causing cellular damage. Particularly, it's very important when it's a neuronal damage. So recently, we started to look into if HHV6 or any other herpes viruses are causing neuronal damage, inflammation in ME/CFS patients, and we tried to look into many different organs, including the different parts of the brain. The study is still going on. We had a huge break because of the coronavirus pandemic, but we hope to start again next month. So we looked into brain of postmortem ME/CFS patients, and we find HHV6 reactivation. We find microRNA U14 in these patients. For example, this is one case in which we find frontal lobe of these patients being infected with having active HHV6 infection. This is another case in which we have also frontal lobe being infected or reactivated HHV6. This is case where we can find upper cervical cord, lumbosacral nerve root showing HHV6 activation. So we have HHV6 activation in patients, but we also see some of the controls which are not classical controls because you don't get tissues from brain that easily from healthy people. So we also see HHV6 reactivation, but some other reasons. So we would like to validate this functionally. This HHV6 reactivation actually means something. It can contribute to ME/CFS. Now, as I told you before, one of the very fascinating result from our in vitro HHV6 work was that when we have viral reactivation in very few cells in a culture dish, the mitochondrial fragmentation could be seen in all the other cells which were actually not reactivating the virus. So how come it is possible that a few cells which are having the activated virus can tell the rest of the cells that something is happening? So what are they doing? What are the factors they are secreting? What are the signals they are sending outside which are telling the healthy cells to fragment the mitochondria? So this data we already published, but I would like to show you a very interesting experiment that we are doing in the lab. So we take a serum or plasma from these ME/CFS patients and the controls, and then we add the plasma or serum to the healthy cells. And then we try to see after 1 day or 2 days what happens to the mitochondria of these patients or these healthy cells. And then we try to quantify this mitochondria. And what we see is that mostly the severe ME/CFS patients, they have their serum causes extreme mitochondrial fragmentation with very small mitochondria. This is particularly a case, a CFS case, very young patients below 20 years of age from Germany who passed away recently. When I took the serum from these patients and added to healthy cells, I saw the mitochondrial fragmentation. I saw very severe mitochondrial fragmentation. But, I did not know what caused this mitochondrial fragmentation. This is something which always puzzled me, that what is there in the serum, what is secreted from the cells, which causes the mitochondrial fragmentation. Yeah. So we still are doing it. We have good success rate, almost 100% success rate with severe ME/CFS patients, but mild and moderate ME/CFS patients, we see a fluctuation because mitochondria sometimes have elongated structure, sometimes a fragmented structure. Quantification we do, but the situation is more clear with severe ME/CFS patients. So what we do in the lab, just to give an idea that it's not a routine diagnostic protocol, which is a simple couple of hours. This is days and weeks of work. So what we do is we take the blood and we prepare a serum out of it, and then we take the serum. We add the serum to healthy cells growing in a petri dish, which has mitochondrial GP protein. So you can see the mitochondria in a live cell under the microscope, and then we grow these cells for 28 to 48 hours. After 28 to 48 hours, we take these cells and put it on a glass slide and fix them, and then we go for a high-end microscopy, this like confocal microscopy. We take several, sometimes hundreds of images from each experiment, and then we go to the computer, do the image processing and quantification. It's a huge amount of work. At the end of the day, we have a quantitative data, how the mitochondria look like when it grows in presence of the serum from patients or controls. Recently, we tried to even look in a different way. So what we did is that we took blood from patients, we isolated the peripheral blood mononuclear cells, or PBMCs, and then we grew these PBMCs in the lab, in culture petri dish. And every 1-2 days interval, we try to collect the media from these PBMCs. So basically, we expected that if any cells in these blood cells are secreting something, there should be some secreted factor in the media. And then we collected this factor and then put on healthy cells and went through the whole entire procedure. And we do see the effect of the PBMC culture media on cells for the very first few days, and later on it gets diluted. So we know that there may be some cells which are short-lived, which are there in the PBMC cultures in the beginning, but later on they are removed because other cells predominate the culture. So we are working on this to solve this, which type of cells are responsible. We hope that we can continue the work. So now coming to the factor, what causes the mitochondrial fragmentation? What is there in the serum that causes mitochondrial fragmentation? We have been working, we have been testing many different type of factors. Trust me, 75% of the time, our work in the lab results in negative results, which we don't talk about, but this is our life. And the 25% of the times, which when we find something interesting, that makes our life happy. So what we have found that a couple of very interesting factors, which are probably something that we would like to continue, and we hope that they are very interesting. One is serum complement activation. We find that ME/CFS patients have a higher serum complement activation. We heard about autoantibodies from Carmen, from Jonas, and I will talk about it a little bit also. So I also believe, and I agree with them, great work from all of them, that autoantibodies probably playing some role in mitochondrial architecture. And an interesting feature also in ME/CFS, what I realized is that there is a large amount of cell death in ME/CFS patients, PBMCs at least, which can cause inflammation to viral microRNAs, human microRNAs, and also through mitochondrial DNA, which is packed into exosomal vesicles, and a very interesting aspect of eosinophil and mast cell granulation also. So why I say the serum complement activation is there in ME/CFS patients, I just want to, this is a busy slide, but I can tell you here that if we take a serum from healthy cases and we treat the serum at 56 degrees for 30 minutes, it's a classical way of inactivating serum complement pathway. You see that there is no effect on mitochondria. So the same cells here, and after the heat inactivation, they don't show much difference. But if you take the CFS patient serum and heat inactivate them, that you mostly lose the effect on mitochondrial fragmentation. This is also done on a large panel of CFS patients and the controls. You can see that the CFS patients, they saw effect on the serum complement inactivation process shows effect on antiviral nature of the serum also. So we believe the serum complement system is overactivated in ME/CFS patients. So we would like to understand this. And in this context, I would like to touch the infection, autoantibodies, and autoimmunity topic, which is being hot topic of discussion today. Carmen talked about it. Jonas also talked about it. And I find their work very fascinating, and Carmen has been publishing on receptors of autoantibodies against alpha beta adrenergic receptors or muscarinic receptors, and they are fascinating from the point of view mitochondrial structure also, because they are, when they are stimulated through epinephrine and norepinephrine, they can also cause mitochondrial structure, mitochondrial fragmentation. And that's why I asked Carmen if I can use their system and their patient-related materials to check if autoantibodies actually can cause mitochondrial fragmentation. So what they do, and what we also would like to do in our lab and start working on it also, is that very simple elaborate process also, that we take the plasma or serum from ME/CFS patients and controls, and then it is being passed through a column which will bind the immunoglobulin G into the column. It can be, there are different type of columns which can bind different antibodies also. So once these antibodies bind to this column, it can be washed, and after washing, with using another buffer, one can take out these antibodies from the column. So we, at the end, we get a rich immunoglobulin G rich fraction, and this fraction is then used by us. We add it to the cells and try to see what happens to the cells. Very interesting. I fully agree with the data from Carmen, from Jonas, that the patients, the IgG fractions isolated from patients with a high amount of beta adrenergic receptor, beta 1 receptor, beta 2 receptor, they changes the mitochondrial structure. But it's very interesting for me is that it's not always straightforward. There are cases where there is a very high amount of IgG, but there is no high amount of beta 2 adrenergic receptor or muscarinic receptor. They also fragment mitochondria, and that's why I believe that together with these receptors, what Carmen has published, there might be some other antibodies which is playing a very important role, and we have to understand this. For example, here is another case in which there are a very high amount of all the four beta 1, beta 2 adrenergic receptor, muscarinic receptor, M3, M4, and then you can see that the mitochondria is fragmented dramatically. It's so brilliant to see the effect of these autoantibody eluates on the cell that you have to believe it, that the autoantibodies are doing something to the mitochondria. But the story is not that simple. So here are a couple of pictures you can see is that if you can follow what I am trying to tell you is that here on the left side, you have a healthy control IgG eluates from a control person, showing a nice fragmented, nice filamentous mitochondria. But once you add IgG fractions rich with beta adrenergic receptor, muscarinic receptor, and you can see that the mitochondria completely changes its structure. You can start to see this mutated mitochondria. And you can see these effects within 16 to 20 hours of adding these antibody elutions from these patients. So it's fascinating. You and discuss about it. And this antibody is against the serum complement protein C1q. So the protein C1 is serum complement protein C1 is a complement classical pathway. It's the first classical pathway protein which leads to other serum complement protein. There are more than 20 serum complement proteins. When serum complement protein C1 is activated, it produces three different types of smaller proteins called C1R, C1S, and C1Q. C1Q is very important for us, and it's very interesting how the C1Q is activated. The immune complexes, IgG and IgM containing immune complexes in exosomal vesicles, they can activate the protein C1 to C1Q. And C1Q has many important roles. I would discuss a couple of them today. One of the key role of C1Q is clearance of apoptotic cells by macrophages. It helps to clean or to clear these apoptotic cells from the body. And if the C1q protein goes down, the apoptotic cells are, the clearance of apoptotic cells are affected, as a result of which autoantibodies against these dead cellular products like DNA starts to appear in the body. Recently, a very interesting paper came out where C1q was shown to control the mitochondrial metabolism of CD8 T cells, which is a science paper, very fascinating paper. So very interesting roles for C1q to play in the body. And there are a couple of diseases, mostly lupus, in which anti-C1q antibody has been shown. And the anti-C1q antibody, what it does is that it binds to C1q and interferes with the function of the C1q. So if you have antibody against the C1q protein, you will have less function of C1q. So the rest of the processes will then be affected. So it results in abnormal response against the pathogenic infections in the body. Very interesting thing is that, which I would like to focus here, is that it has been shown that the EBV infection, after the EBV infection, one of the protein or a peptide from EBV, it helps in generating anti-C1q antibodies in the body. And the antibodies generated against these EBV protein, this is called molecular mimicry, and as a result of which the anti-C1q then starts interfering with the function of C1q. So EBV indirectly can produce autoantibodies against the C1q, which is being documented. And another interesting factor for me is that it's an argument why we should study on these autoantibodies of C1q, is that when we do exercise, there are a lot of publications that when we do exercises, hectic exercises, what happens is that the C1q levels goes down. So my argument is that if the in ME/CFS patients, the C1q protein is already at a threshold, a very at a level where a little bit of deviation in the C1q levels can make a big difference, and patients do exercise, it just might be possible that it goes beyond the critical level and creates a lot of clinical features that we know is a classical features for ME/CFS. So my interest is in anti-C1q antibodies and a couple of other functions of the C1q protein, which you might be interested in knowing. For example, is clearance of pathogens. So C1q protein binds to bacterial pathogens and it helps in their clearance. So if you have interference with C1q protein function, then you will have unusual bacterial infections also in the body. It also regulates the dendritic cell maturation and function, antigen presentation of TLB cells. So if you have interference with C1q levels, all these functions will be affected. In the brain, C1q is known to be both neurotoxic as well as neuroprotective roles. A very, very tight balance of C1q is important because it controls the synaptic pruning and plasticity. So if you have imbalance in the C1q levels in the brain because of autoantibodies or any other reason, you will have effect on central nervous system. And another interesting feature, I also recently asked people in Twitter that, is there any problem with Undhealing? We heard about endothelial cell function from Carmen also. C1q in endothelial cells, it induces angiogenesis. It helps in blood vessel production and Undhealing. So it has been shown that in a C1q deleted mice, there is always limited number of blood vessels. So this is a very interesting feature of C1q protein, and I believe that C1q protein activity goes down in ME/CFS patients, at least in a group of ME/CFS patients. So if you want to learn more about these functions of C1q, I will recommend to read this Frontier Immunology review article. Now, I talked about lupus. Lupus is one disease in which anti-C1q antibody has been well documented. Other diseases are not that familiar, not that well studied. I took a picture from a JCI paper over here. I would like to focus on, this is a busy slide, but I would like to talk about a very interesting aspect of this. So when these authors, they injected the autoantibodies against C1q in a mouse, wild-type mouse, they did not see any lupus-related effect. But when these mice were pre-injected with a another antibody against a GBM protein, and then later on the mice was injected with anti-C1q autoantibody. They started developing lupus. And when they took a lupus-prone mouse and they injected the autoantibodies, they have a complement activation, apoptotic body clearance was decreased, and then decreased availability of the C1q to clear the apoptotic bodies. So all these features are fascinating, and that tells us that probably the autoantibodies against the adrenergic receptor, muscarinic receptor, has a role to play together with another autoantibody, maybe it's anti-C1q antibody. So to do that, we did a small cohort of samples. We checked the level of anti-C1q antibodies. We established these assays in the lab. When corona pandemic was going on, we were sitting in lab 4 or 5 hours a day and establishing these assays. And we found, here you can see that a large number of, or rather a group of ME/CFS patients have very high amount of anti-C1q antibodies in their serum. We do see non-ME/CFS patients also having few cases having high anti-C1q antibody, but interesting is that these are classically non-ME/CFS patients, but these are EBV infected or active EBV having patients. So EBV infection do increase the anti-C1q antibodies in these patients. We had a possibility to check a couple of post-treatment Lyme disease patients, and we did not see anti-C1q levels to be high at this moment. What we did is that we tried to see, okay, we have anti-C1q antibody high in serums, but does it do something to the mitochondria? So we threw these antibodies, 20 units of antibody per ml into cells. I took 20 units, one picture over here, because you see that the general or the normal level is around 10 units per ml, and 20 units, many patients have more than 20 units. So I took 20 units of anti-C1q antibody and put on healthy cells. And in 12 hours time, I can see mitochondrial fragmentation in these cells. They are extremely strong in mitochondrial fragmentation. And this is something which is fascinating for us, and we would like to continue working on this. Hopefully we can do this. Another aspect that I would like to talk about is serum complement activation and high amount of a protein called eosinophilic cationic protein levels in ME/CFS. These proteins are generally produced by eosinophils as well as the mast cells. We heard about mast cell activation today. So all these cells, when they are granulated, they're activated, they produce a large amount of eosinophilic cationic proteins. These proteins are well-documented roles or high levels in allergy patients. Particularly, we know that ME/CFS patients have many different types of allergies against drugs, against food, and other things. So I personally believe that there is something to do with this, because these proteins are cytotoxic ribonucleases. If they are produced in extremely high amount, they can cause a cell death, which is something which we see in many ME/CFS patients, not in all. It's a small subgroup of patients, but we see that if we take the serum or plasma from these ME/CFS patients and you grow cells, the cells die. So it's something very interesting, and we recently started to do the ELISA assays to look into ECP protein levels in patients, and we do see ME/CFS patients having higher amount of ECP levels. Interestingly, there is an old paper which also shows that ECP proteins are high in ME/CFS patients. So I'm not the only one who is telling about it, or not the first one. So it's very interesting, and we would like to look into ECP proteins, what they do to the cells and the mitochondria in future. To summarize then at the end, that at this moment we have 3 very interesting serum transferable components that we would like to characterize at molecular level in our lab. We are not clinical people. We are not dealing with the patients directly. We cannot treat patients, but our job is to try to see what is seen in clinics. Do they have direct relevance to ME/CFS at the physiological level? So we are trying to look into autoantibodies that effect on mitochondria, anti-C1q autoantibodies is something which fascinates us at this moment. We would like to see serum complement activation. It's very interesting because serum complement activation is related to C1q also. And we have we are looking into exosomes and mitochondrial DNA, their role. It's also very interesting. This we are doing with Bob Navio. So at the end, I would like to show some results about what we are doing in recently. It's not what we are doing. You know that pandemic is going on, SARS-CoV-2 infection, and a lot of patients have prolonged symptoms, which is similar to ME/CFS. So a lot of talk is going on, discussion is going on, and this fascinates me that how SARS-CoV-2 can cause ME/CFS or ME/CFS-like symptoms. I am fascinated particularly with the delayed appearance of the symptoms. So I believe that it is not the SARS-CoV-2 which is causing the ME/CFS-like symptoms, rather the ME/CFS after the SARS-CoV-2 infection, directly or indirectly, use of drugs or other things, we actually reactivate herpes viruses, particularly HHV6, EBV, and things like that, CMV. And these are neurotropic viruses. And once these viruses are reactivated, they cause complications, inflammations, and things like in the brain, which leads to ME/CFS-like symptoms, which fits to our hypothesis. So we would like to know that if SARS-CoV-2 has a direct effect on other herpes viruses. So we recently started doing infection experiments because of very active work from a lot of people in our university. We have now the SARS-CoV-2 in culture. So what we did is that, for example, this is just a preliminary experiment I would like to share. We took cells with latent HHV6 and then we infected with SARS-CoV-2 for 1 to 2 days. And then we looked into mitochondria of these cells as well as what SARS-CoV-2 is doing inside. At the same time, we prepared RNA and we do transcriptomics, single-cell transcriptomics to see what happens at the RNA level to these cells after the SARS-CoV-2 infection. These are a couple of pictures, no data to discuss over here, but just to show that it's very fascinating. So here, cells with the mitochondria. So we are trying to see what happens to the mitochondria after the infection. Very interestingly, the cells which are reactivating, so it's cells with green are reactivating cells, that the virus is reactivating, and you can see the SARS-CoV-2 is sitting in these cells or surroundings. So it's very fascinating. It's just preliminary, but at least it gives us a possibility to look or to try certain hypotheses and to see if SARS-CoV-2 will have a long effect. And probably, if we are right, then we probably can find solutions to this problem. This is another picture just to share. So I believe that together we can, and we will fight ME/CFS, but definitely it will take time and it needs a lot of effort. So thank you very much for your attention. I hope that I gave you something to think about for the rest of the day, for the rest of the week, for the rest of the month. Thank you very much for your attention, and I would love to take questions from you.